Thanks for joining me today. Appreciate your time. HypoxyLab system is an incubator for cell culture, an incubator that supports a controlled oxygen environment, as well as a simultaneous manipulation in that environment of cells in culture. So generally, the term used for a product such as this is a hypoxia workstation. I wanted to just start with a very brief bit of background on the company itself. Oxford Optronix was founded all the way back in 1991 by the person that remains our CEO. We're a small company. We've always been and are still based near Oxford in the UK. And although we're a small company, we're very proud of our rich heritage of unique scientific instruments that we've developed and that we manufacture here in the UK. And on that journey and in that portfolio of products, our most recent product is in fact the HypoxyLab system, which I'm going to be talking to you about today. And I guess while by definition, I'm assuming that most people joining this webinar have an intrinsic understanding of the relevance and importance of oxygen in physiology, I thought I'd just make a few general points, which is probably general knowledge anyway. But obviously, as we all know, oxygen is essential for cell survival. That's very basic. In the living in vivo state, so in living tissues, the availability of oxygen at the cellular level in the microenvironment of each cell is essentially a balance between oxygen delivery by the blood and oxygen consumption by cells in that microenvironment. And that oxygen availability, the concentration of available oxygen to cells, has a very profound effect on the metabolic status of cells, their activity, their growth characteristics, as demonstrated by a very large body of peer-reviewed scientific evidence. For instance, most notably and most famously surrounding the HIF1 alpha cascade pathway, the master regulator as it's sometimes called, work which has even been the subject of a Nobel Prize back in 2019. And in fact, one of the named professors on that prize is an Oxford-based professor whom we know and have known for many years ourselves here at Oxford Optronix. But yeah, that Nobel Prize, I guess, was a fantastic vindication of the importance of the cellular oxygen environment. Okay, so I guess I wanted to just go a little bit deeper into what is physiological oxygen. What kind of oxygen concentration is typical in the in vivo state? And so I thought I'd just summarize kind of some typical values. Now, these are generalizations. If you look these data up in the literature, you will get a range. So these are typical values. And in air, so in the air that we breathe at sea level, 21% oxygen, that's approximately equivalent to 160 millimeters of mercury. So we at Oxford Optronix, and I'll come back to this in a minute, like to describe oxygen in partial pressure, not in percent. And that's a crucial distinguisher that's relevant to the HypoxyLab. And I'll come back to that too. By the time we breathe in the air and the air gets into our lungs, deep into our alveoli, if you look at the cells that line our alveoli, they're seeing approximately 90 to 100 millimeters of mercury of oxygen concentration. If you look at the arterial blood, so the blood in our arteries, that's typically 75 to 90 millimeters of mercury. This is the oxygen concentration. Venous blood, 30 or 40. And if you look down at the tissue level, so by the time you get to the microvascular beds, the very far reaches of our microvascular system, the tissue itself is seeing somewhere in the range of 10 to 50 millimeters of mercury of oxygen concentration. Obviously, it's variable. Like I said, it depends on the rate of oxygen supply versus the rate of consumption. Tissues are different, their metabolic activity levels vary, and so there are lots of factors that affect the oxygen concentration in any one in vivo region, any one region of tissue at any one time. But it's approximately 10 healthy levels are approximately 10 to 50 millimeters of mercury, which, as you can see, is approximately, well, at best, a third of the concentration of oxygen that we breathe in. And this is an important point, because cell culture typically around the world is still carried out in standard incubators that provide a 37-degree environment, a humid environment, and a little bit of CO2 as a buffer, but do not control oxygen at all. So the vast majority, to this day, the vast majority of cell culture occurs at room air, so 21% at ambient levels of oxygen, which is clearly in no way physiologically relevant. It's way above typical physiological levels and potentially has all these metabolic effects, and so risks affecting the very pathways and data that the scientist is looking to generate. So I guess I was wanting to refer to that as a cell culture disconnect. So while there have been devices that are, and incubators that are capable of controlling the oxygen environment, including workstations. We refer to the HypoxyLab as a workstation because it not only can create the environment of an incubator plus control the oxygen, but it also allows manipulation of cells in culture within it, hence the word workstation. There have been workstations, hypoxia workstations, on the market for some years now. However, they tend to be very large devices, comparable in terms of footprint to laminar flow hoods, that kind of thing, and requiring dedicated lab space. They're often sited in cell tissue culture rooms. So some years ago, we decided that there was a need for an equivalent system that was much more compact, potentially much more efficient at using gas, consuming gas, and also just generally easier to use and more flexible. And hence we came up with our HypoxyLab product, which, as you can see behind me, if you can get an idea of scale relative to myself, is intended to fit on a standard laboratory bench. Sure, there's a little bit of altitude needed, but in terms of depth and footprint, this system does fit on a standard laboratory bench. And it is suited, just like any incubator, to the continuous culture of cells. It is, just because it is capable of supporting manipulation internally of cells, and because it is capable of controlling the oxygen environment internally, doesn't mean that it is any less capable of long-term cell culture, just a point. Okay, so in terms of the benefits of the system, there's a few key ones, which are, I guess I'm repeating myself, but just to re-emphasize, this is a system that will maintain within it a controlled environment of temperature, CO2, humidity, but crucially also oxygen levels. It's compact enough to fit on a laboratory bench. Its size also means that in terms of gas consumption, in typical usage, its reduced physical volume compared to the more common, very large systems is dramatically less. And obviously that has long-term benefits in terms of the cost of supporting and running the system. And we've done a lot to make the system very simple to use. And I will come back to that when I turn around and physically show you around the system in a minute. But before I do that, there is one key point which I wanted to come back to, and that is my reference earlier to the fact that we like to talk about oxygen concentration in terms of partial pressure rather than percent. And that is a key differentiator between the way we approach oxygen control in the HypoxyLab and what seems to be the industry standard. And that is that we use and we control the environment, the oxygen component of the environment inside the HypoxyLab, using a measure of partial pressure of oxygen not just oxygen percentage. And that's important. Now, this next slide explains this, and I don't want it looks complicated, and I don't want to go into too much detail. I'm happy to answer questions afterwards, but just to demonstrate, fundamentally, the advantage of controlling the oxygen environment using partial pressure is really boils down to the fact that our system, so the HypoxyLab, intrinsically compensates for fluctuations in atmospheric pressure. And that fluctuation can either be the result of weather changes, low pressure systems, high pressure systems, those affect the overall atmospheric pressure. And if a chamber is being controlled using just the percentage of oxygen, that's not protecting the cell cultures from oxygen concentration fluctuations, whereas using partial pressure of oxygen does. Similarly, laboratories that may be situated at altitude. Now, obviously, that's relatively unusual, but there are two cities that come to mind: Mexico City, Denver in Colorado, the US. Those are both cities at significant altitude, and there the mean atmospheric pressure is considerably less than at sea level. And again, unless you're using a hypoxia workstation that is being controlled using the partial pressure of oxygen, you are not compensating for that atmospheric pressure difference. That was very long-winded, I'm sorry, but just to go over these numbers real quick. At sea level, we breathe in 21% oxygen. And if you do the calculation, that's in terms of millimeters of mercury, barometric pressure, that's 760 millimeters of mercury, of which, as we said, 21 or 20.9% is oxygen, which works out as 159 millimeters of mercury. So that's the standard partial pressure of oxygen at mean sea level. You go up by 500 meters, you climb a mountain, the atmospheric pressure drops typically to about just over 713 millimeters of mercury, of which 20.9% is only 149 millimeters of mercury. You go up to 1,500 meters, and that's 131 millimeters of mercury there straight away. So you can see that altitude has a very significant effect, and so does atmospheric pressure. So do atmospheric pressure changes as a result of weather systems. And the key point is that by controlling the oxygen environment using the partial pressure of oxygen, as you can see, we can read that fluctuation and we can compensate to maintain a constant partial pressure of oxygen. And it's the partial pressure of oxygen that is representative of the oxygen concentration that cells in culture see, or cells in our bodies see. So that, in short, the key point I wanted to get across is that we are controlling the oxygen environment in a different way, in a way that we feel is much more scientifically rigorous compared to the existing systems on the market. And I guess just a bit of background. The reason we do this, actually, and I meant to mention this at the very beginning, we've been in business since 1991, and one of our long-standing core competences and product ranges has been a dissolved oxygen monitor. So we've been working with devices that measure oxygen in partial pressure for like over 25 years, getting on for 30 years now, which is why we understand the importance of partial pressure of oxygen, which is why we chose to control the environment using that unit rather than percent oxygen. Okay, I'm going to skip the description because I'm going to show you the device directly in just a second. But before I do that, there was a couple of add-ons I just wanted to describe. Again, I have them here and I could briefly show them if you like. But I just mentioned our oxygen monitors. That range of systems is called the OxyLite. And the HypoxyLab is compatible with OxyLite in that the OxyLite can be used to generate a measurement, a direct measurement of dissolved oxygen, which is another way of saying partial pressure of oxygen, dissolved oxygen, within cell cultures, directly from inside cell cultures, or even bottles of medium that are inside the chamber. So if you as a user require the ability not only to set the environment to a particular oxygen concentration, but also to actually measure directly from your cell cultures what is the actual true oxygen concentration that my cells are being exposed to, then you can use one of our Oxylight devices and an optical sensor, which I can show you in a second, to make that measurement. Actually, I think the next slide the next slide shows it slightly closer. This is a view of the optical sensor. It's a miniature, literally a fiber optic, onto the very tip of which we attach a bit of chemistry. So the oxygen measurement can be made from the very tip. And what you're seeing here is a little port, a little gland, as we call it, situated on the side of the system that allows you to pass the sensor into the chamber, seal around it, and make measurements from the inside. So the monitor, this monitor sits on the outside, or on the image there on top, and it's the sensor that's passed in through a port on the side. The other device, just briefly to touch on, is the CytoSmart, and that is a miniature digital microscope. So if there is a requirement to be able to take images of or view in real time a magnified view of cells in cell culture within the system, then the CytoSmart is a device that is intended for continuous use in high humidity environments, so in incubators. It can be left inside the system for very long periods of time, and it connects to a PC on the outside via a USB through port that we have built into the system, and I'll show you that in a minute as well. So two accessories, if you like, add-ons that we optionally offer with the HypoxyLab. Okay, so I guess that kind of pretty much brings to an end the few slides that I wanted to share with you, and I'm going to transition now to showing you the actual system. I'll try and bring the camera a little bit closer. I could potentially pick up the camera and show you elements more closely. I am just conscious of not taking up too much of your time and allowing things to get boring. But on this slide, just to point out that I've made a little list here of optional further reading. If anybody's interested in reading reviews, there's a mix of reviews here and articles that talk about from various angles the relevance of oxygen in tissue culture or in cell biology generally. But the other emphasis is on simplicity. So we've done everything we can to make the system as simple to use as possible. And I can demonstrate that, for instance, by showing you how easily the whole top just comes off. So if, for instance, you need to clean the inside, or you want to prepare the system for startup and you want some large items inside, the whole enclosure cover just lifts off. And let me show you that real quickly. Although actually, just before I do that, let me just go back to basics. Obviously, I said this is an incubator. It doesn't look like a normal incubator, and that is because it is intended to allow users to insert their arms through these two arm ports to manipulate cells without removing them back into atmospheric conditions of oxygen. So it is a workstation, and that's also why it's clear, why you can see into it. There's a vision panel here, but the whole system is actually clear. So yeah, just to point that fact out, there isn't a great big silver door as there is on a bog standard incubator. So just to demonstrate that the whole system comes apart very easily. So this whole cover just lifts off, and that actually gives us a better view of what's going on inside. Hopefully you can see that okay. We have some lights that we can turn on up there. We have space, obviously, for cultures, flasks, petri dishes, and I was just going to demonstrate that these shelves are adjustable, can be removed, and also to point out that they are designed with all these holes for two reasons. A, to ensure that the atmosphere inside can move freely underneath any samples that are placed on these shelves. But these shelves are also compatible with 50 milliliter Falcon tubes and also the smaller 15 milliliter Falcon tubes. And they can also be used to hang and suspend like pipetters and things like that. So very easy, simple to adjust and reposition. And then I wanted to just briefly point out, I don't know if you can see that just about, there's a little hole here, or a little cap. And that is the USB through port for the little miniature microscope I was showing you just now, the little CytoSmart device. Comes with a little USB port, USB lead, that connects to this connector on the inside, and a port on the left-hand side to the PC that allows you to view the output of this little microscope. What else did I want to show you? Oh, yeah, there's one little thing, which I don't think I mentioned. One of the things that's built into this device is a HEPA filter. So the environment, the internal environment of the HypoxyLab is permanently filtered through a HEPA filter. And that HEPA filter can be exchanged very easily. It's attached to the rear of this turret structure that's inside the HypoxyLab. And it's just an exchangeable consumable that's very cheap. It's something that we recommend is changed approximately once a month. But beyond that, probably, okay, there's, yeah, the other couple of things that I wanted to show you quickly that are relevant to the simplicity of use of the system is the entry door. So we have like a little entry hatch which can be used to pass items in and out of the system while it's in use. So let me just return the enclosure cover back onto the base. Okay, and I'm going to switch the system on in a minute, but just to demonstrate that, first of all, these arm ports, these are sleeves with latex cuffs that are designed to seal around the wrist. And in use, of course, what you would do is you don't want to be leaving these open. Of course, you don't. The point is that we're maintaining a low oxygen environment here inside the system that is equivalent to normoxia, to physiological oxygen levels. And of course, you need to avoid or prevent atmospheric oxygen getting inside. And so these arm ports are typically plugged using a little arm port, wrist seal plug such as that in use. But to put the arms inside, you would remove that briefly, position your arm inside, and now you can use a pipette to manipulate cells, passage cells, harvest cells, whatever you need to do. There's space to do it. And the point is that the oxygen environment, the physiological oxygen environment being maintained is not being broken. So the cells aren't being removed back out of the system into atmospheric oxygen. The idea is that cells can be maintained within the system over the long term, medium or long term. The other thing to show you is that we use a very simple approach to allow users to pass things in and out of the chamber during use. Now, again, this is where we differentiate ourselves from the more traditional large systems. Those will typically have what's known as a separate kind of add-on transfer hatch through which everything that needs to go inside, in and out, has to pass. Now, there's a reason for doing that, because it's to minimize the amount of external oxygen that can get into the system. But in fact, what we've done is to devise a very convenient letterbox kind of system, which is easy to open. So even while the system is in use, this door can be opened. There is a secondary, and I don't know if you can see that, a secondary clear flap on the inside. So even with this door open, there is virtually no way for external oxygen to get into the system anyway. But if, let's say, you did want to pass something into the chamber, you would just pass it under the door into the system and then make sure the door was closed quickly afterwards. In addition to that, the system recognizes when this flap is open and immediately starts injecting some nitrogen to generate a little bit of positive pressure that very much minimizes ingress of any atmospheric oxygen. So the effect on the internal environment of passing something in and out of this flap is essentially nil. And yet this is a much easier and a more convenient system to use than the transfer hatch mechanism that is more typical of the larger systems that I'll keep referring to. So there's a few aspects relating to the simplicity of use that I wanted to point out. Beyond that, there's a couple of things I wanted to talk briefly about how we maintain the environment inside. So we generate humidity using a nebulizer-based device. So it's active humidification. It's not just evaporation-based, which is what's typical in incubators, in standard incubators. So we actually inject humidity via a nebulizer-based device that's built into the base. And in terms of gas supply to the device, you need CO2, just like for any incubator. The typical cell culture concentration is 5%. That's normal. But there are two further gases that need to be attached in the case of the HypoxyLab in order to allow it to maintain any desired oxygen concentration. And those are nitrogen, pure nitrogen, So effectively, we reduce the internal oxygen concentration by pumping in nitrogen and displacing oxygen, really. But we also need, and maybe you can see there are three gas lines here. One is CO2, one is nitrogen, and the other is a supply of air, actually, synthetic air, because it might be that you're at very low oxygen concentration, but you want to increase it a little bit, in which case you do need to inject a little bit of air. It's not atmospheric air, it's pure air supplied by a gas bottle, so it's not contaminated, but the system needs those three gas supplies to function, as well as a source of deionized or distilled water for the nebulizer. And if you like, I can show you what things look like at the back, where everything connects, in a minute. But before that, I think I wanted to just give you a very brief introduction to the touch screen. And again, we've designed the system to be as simple as possible to use with a straightforward touch screen interface. And maybe I should try and show you that in greater detail. Again, I want to try not to spend too much time on this, but I just wanted to start the system up and give you a feel for how that looks. So I'll press start. The system takes approximately, I think, 40 seconds or something to boot up. And you can already see along the bottom here, these are my primary parameters. And let me see if I can grab the camera, hang on, and I'll bring it a bit closer. And hopefully, if I can line that up, you can get a better idea of what's going on. So we have all the primary parameters along the bottom here: oxygen, CO2, temperature, and humidity. And these are the points where I can set the desired levels of each. On top of that, we have, let me just reduce the scale, hang on. On top of that, we have some traces down the center. Can you see that okay? So we have a real-time measurement of oxygen. Is that readable, by the way? For me, it's like a mirror image. What's it looking like for you? It's readable? Okay, perfect. Yes. So we have the oxygen concentration, which at the moment is showing about 130 millimeters of mercury, because it's already coming down from atmospheric levels. We have a real-time CO2 concentration and the real-time temperature. Obviously, that's at room temperature, and it's going to take some time to warm up. And down here are the parameters that the set points, as we call them, that I wish the system to reach. So in terms of oxygen, I have set a desired set point of 38 millimeters of mercury. Hopefully you can see that okay. And so if you do the calculation, that's approximately equivalent to 5% CO2, sorry, 5% oxygen. And so this would be a very typically experienced kind of physiological concentration or partial pressure of oxygen. And as you can see, if I look at the trace, the oxygen is already coming down. We were at 130. It's now at about 107, 8 or 6 and dropping. CO2 is coming up. So I've set CO2 to 5%, which is the common parameter, the common concentration used. And as you can see, it's overshooting just a little bit, but that's come up quite quickly. And then, which is a little bit harder to see, but the temperature is gradually rising, and it's set obviously to 37, which is what pretty much everybody uses. And in terms of humidity, I can also set the humidity. At this point, it's set to 7. Again, I'm not sure that's focusing very well. Apologies. But that's set to 70%. And it goes up to 85% is the highest level that's possible. Yeah, so as you can see, very intuitive, simple user interface. The amount of trace data displayed at any one time can be controlled here in the bottom right corner. So I can make that 10 minutes of data or 20, all the way up to, I think, 24 hours of data. In fact, 48 hours of data if necessary. And then back to 1 minute, 2 minutes, 5 minutes, whatever is desired. Yeah, so there's all sorts of other functionality, which I think maybe is not relevant today, but my main message is that it's a very simple, intuitive user interface. Okay, so while that system continues to run, oops, let's do that. I guess there's one more little thing I could show you, and that is that for long-term incubations, we do recommend the use of a jacket, a thermal jacket. We have a thermal jacket available for the system, which looks like this. Just a second. It's essentially like a sheet of, it's like literally like a jumper for, it's the same material as like a wetsuit for diving. So it's stretchable and it literally fits over the whole system. And we offer that for two reasons. One is to reduce the power consumption of the system because it offers a thermal insulation. But the other reason actually is to minimize internal condensation from the high humidity levels that are typical in an incubator. Because we need to be able to make sure that you can see into the system, the walls are quite thin, but they're not insulated. And depending on the external conditions, if the internal system or environment is at 37 degrees at a high humidity level and the outside lab temperature is quite cold, you do get some condensation on the inside, but that can be almost eliminated using that thermal jacket. So that's an accessory that is included with every system. Yeah, so yeah, conditions are continuing to reach set point. Typically, we could suggest that you turn the system on about 30 minutes or so before being ready to use. So it takes about 30 minutes to reach a steady state. Primarily, the limiting factor is the rate at which temperature reaches 37 degrees. The remaining parameters reach steady state more quickly. But yeah, that's it. The system will just maintain whatever levels are set. One feature I guess I could just briefly point out is that and I don't think you need to see this up close, but we have an optional feature here that allows a user to pre-program a profile of oxygen level. So this is mostly relevant to scientists working in sleep apnea, where there's a desire to expose cells to different levels of oxygen for different periods of, so for example, 40 millimeters of mercury, then very hypoxic levels, 10 millimeters of mercury, then back to 40, then 10 repeatedly. And that can be pre-programmed and the system will automatically switch between those gas levels over the course of whatever, 24, 48 hours. So that's an additional little feature. But yeah, I think that's basically it. I don't think it's necessary to show you the rear unless somebody wants to see. I've also got some more images I could show depending on questions, but I think I'm just checking my notes here. I think I think I've pretty much covered everything that I wanted to cover, and I'm happy to answer any questions if there are any.
Yes, we have.
Great.
Robert, you want to read the question?
Yeah, it's okay. I may read it. It's one question from Penelope Aguilera, which is, how long does the chamber take to reach the oxygen concentration required? For example, 1%.
Perfect. So like I was saying just now, the limiting factor is probably, and mostly the limiting factor is the time taken to reach 37 degrees, which is about 30 at very low oxygen concentration. So 1% is approximately equal to 7 or 8 millimeters of mercury. That'll take about the same amount of time, actually. If you set it to even lower concentrations than that, so 0.5%, that can maybe take 40 minutes, approximately.
Okay, just checking out if there's any other question on the YouTube channel. And you got any, Alice?
Yes, I have a question, Michael. Some research think that the system is more than they need, no, or too large for their needs in this moment. What can say to you to this research for use?
So when you say it's more than their needs in terms of functionality or in terms of?
Yes.
Okay. Yeah, I guess what we have designed or intended to create is a significantly lower entry point into the ability to carry out hypoxia work, if you like, or to carry out cell culture under physiologically relevant or physiologically representative conditions. We believe that this HypoxyLab makes it a lot more accessible for many more scientists than ever before. There, long term, we are thinking about whether there is scope for an even simpler system, an even smaller entry point. That is a possibility for the future. But at the moment, unfortunately, this is all we can offer. But the advantage is that it is fairly compact, it is fairly flexible, and it's relatively mobile. It can be picked up, it can be put on a trolley, moved around. It's easy to set up. We are finding that, at least for a lot of people, this is unlocking the ability to do that kind of work, which wasn't an option before, when all that was available are these huge systems. Oops, my chair just collapsed. What happened there? I suddenly disappeared. It's because my chair. Yeah, we can't cover all the bases, but I think we've made that kind of work much more accessible to a lot of people. But yeah, we still think there's maybe more work or other interpretations of our technology which would allow us to go even further in the future.
Oh, thanks, Mike. And another question, I think it's very simple, but in Mexico, I think it's very important, this question. What is the difference in use anaerobic cabins or with HypoxyLab versus HypoxyLab?
Yeah, no, that's a great question, actually. Yeah, because anaerobic incubators and workstations have been around for a long time, actually, to meet the needs primarily of bacterial work. And that's the difference, is that anaerobic chambers are intended to generate a true zero oxygen environment for microbiological applications where oxygen is highly toxic. But what they don't do is allow you to maintain a low oxygen concentration. So the key difference is that anaerobic chambers will allow you to generate a zero oxygen environment, but nothing above that, no in between. It's either atmospheric, it's switched off, or when you turn it on, oxygen drops to absolutely nothing. Whereas for mammalian cell culture, what's relevant is that kind of 10 to 50 millimeter mercury range that I was talking about earlier. And this system allows you to set anything between 1 millimeter mercury all the way to 100 and can't remember what it is now, 120 or 130. So approximately 18% oxygen equivalent is the highest. And that's the key difference, is that you can set any concentration you want, not just zero.
Oh, okay. This is the most important.
Yeah. So I guess it's a difference between the needs of microbiologists working with bacterial cells, yeast cells maybe, and people working on mammalian cell culture.
Thank you, Michael. I don't know if are more questions, Robert?
Just let me check. There's one on YouTube, one comment from Luis Sobrevia, which is nice demonstration, exciting equipment, any information of potential variations in the O2 concentration, low or high, for cells in serum-free medium. Any difference with cell in media containing serum?
I don't think so. I believe we have users that use the full range of cultures with or without serum. There's no reason why that will affect the ability for the system to ensure that cells are exposed to a reduced physiologically relevant level of oxygen. If I've understood the question correctly, I don't think there's any effect on the ability for the system to maintain cells at a desired oxygen concentration with or without serum. I don't think it affects things.
Yeah. Okay. There's another one from Penelope again. Are there replacement of all the pieces of the chamber?
Yes. So there are various consumables or various parts that need to be replaced from time to time. That's a good question. For instance, the HEPA filter, which I showed you earlier, there is also a seal all around the base that ensures an airtight seal between this part and the base every time the thing is removed. That is a silicone material that needs to be replaced from time to time because it wears out. Similarly, these latex wrist seals, they can be replaced and they're available as free or as accessories, consumables effectively. Yeah, I guess again, it's all about seals really. The seal around the door, that can also be replaced. We do recommend, because the system is operating at 37 degrees in a high humidity environment, there is some wear and tear over time. We recommend annual servicing of the system, which is not a very complicated process, but it's something that we offer on site. And if it's through our distributor partners, we provide the training and instructions to allow your distributor to do that work. It's maybe 2 or 3 hours of work once a year. That's our recommendation to ensure optimal performance of the system in the long term. But yeah, all parts are replaceable. Also these sleeves, these white sleeves, anything really, even the whole enclosure cover, if for some reason it fails, which is never done, but all parts, yeah. Okay, I think, am I right in understanding the question, what is the difference between this system and a tri-gas incubator? Well, that's a good question. Fundamentally, really the main difference is the fact that with this system, you can manipulate the cells while they're in culture. Whereas with a standard tri-gas incubator, you put your cells in, you close the door, and while they're inside, they're being exposed to your desired oxygen level. But as soon as you want to do anything with them, you want to harvest them or whatever, you need to get them out. And as soon as you get them out, you're exposing them to an oxygen shock straight away. Whereas in here, you can do the manipulation and do the harvesting in that same oxygen environment before removing them. So I guess that's the key difference.
Okay.
The fact that this is a workstation that not just allows you to incubate your cells, but also to manipulate and harvest them inside, internally. Plus, I guess there's a few other benefits, is that you can you could set the oxygen level much more easily. What I haven't mentioned also is that these data can be recorded continuously. In fact, what we generate are files that are compatible with your lab chart reader software, actually.
Exactly.
And that's accessible via a USB stick at the rear. So that, yeah, there's all sorts of other functionality and the ability to move stuff in and out without opening the door. Every time in a standard incubator, every time, let's say you want to add, somebody else wants to add some cells, and then suddenly you're opening the door wide, the whole inside comes back, potentially quickly climbs back up to atmospheric oxygen, then you close the door again and it has to come back down. Whereas in this case, you can move things in and out without disrupting the environment.